No, we just like, not even like doing that.
No rush. I just want, that was my, kind of my contribution.
My mom made it. Let me just go ahead and just say something. Sometimes
Because I.
No.
Yeah.
Yesterday. All right.
So we're trading dad jokes.
Uh-huh.
What do you call a dog in the winter?
So.
What?
I think.
Chili dog.
Oh, that's cute. I like that.
Saturday, I had to go back to my dad's blood pressure. My stepmom had to die. You know what? I would take care of him. My stepmom.
Oh.
I was, my parents were talking about that.
Okay. Okay.
I don't know if I'm gonna you can walk there out of my house. I just went out alone.
Go to bed and feel like reaching out. The doorbell
And
was
I
playing.
catch up, my ready for today. I will get.
Okay.
I don't know.
And I'm scared to.
Yeah.
Where did the pen go?
Right.
I'm gonna go on.
Yeah.
I don't want to mention.
Right.
It's just in this, like, why is it?
Wow.
Manage the Kahoot. While you guys are getting signed into the Kahoot, wish I could have this up and pull up the other thing I need to see. I can't. Okay, cool. All right, so I have this written on this board, but it's clearer on the thing here. This is what we have left until fall break. So basically until our midpoint for the course. Today is Tuesday, September 30th. We are doing chapter 9, 25, and 10 during the lecture time. And lab will be doing lab 10. It's a lot of talking, lab 10 is. There's not a lot of doing on your part, but there's a lot of content that we'll be covering on lab 10. Obviously, the pre-labs kind of indicated that. We had 4 pre-labs. And so we're going to be talking a lot about that content. We're not doing PCR. We're not doing gel electrophoresis. We're not actually doing the DNA stuff because they take hours and days. And so we just, screw it, right? We can talk about the content and then cover that stuff. You guys have to know the concept for the exam. So we'll be talking through that stuff. And I'll also give you guys a little bit of opportunity because it relates back to chapter 6. If you need me to clarify anything for chapter 6, I hate to bring it to you, but when we talk about transformation, basically the way that our genes are regulated in our plasmid when we put them into our bacteria or to the transformation lab for lab 10, the genes are regulated by an operon. It's an inducible operon. And so again, y'all thought we would be done talking about an operon? Forget that. Like we keep bringing up a lot of our concepts over and over and over again. And if we aren't picking up what the things are, then we're not getting what we need out of the interaction. So there's another opportunity for us to talk through operons as well as all basically chapter 8, which we did get to discuss a lot in class, really that much. So we'll be talking about that stuff in the lab. Because it directly applies to the lab stuff we'll be doing in lab 10 today. Then on Thursday this week, obviously we don't have lecture on Thursday. Lab 11 will be on Thursday. That's our ELISA lab. Don't worry about it till we get to it. And then next Monday, we will be doing chapter 13 and 14. We still have, lecture that week. So FYI, chapter 13 and 14 next Tuesday. Excuse me, you guys are Tuesday, Thursday. Tuesday next week, we will be doing chapter 13 and 14, and then also doing a lab review that week of that Tuesday, the 7th, next Tuesday. You can technically join via Zoom on both of those days. It should be Tuesday, the 7th for you guys, okay? You can join via Zoom for both of those. Attendance is still required for both of them. It's not optional attendance, but you can join via Zoom. And lab, again, not optional attendance. It still counts as a lab, participation day. So if you miss it, it counts against you as one of your absences in lab. So just remember that. But it's basically, you have to join them, but you can join via Zoom. I will send out an email probably on Friday this week, just reminding you guys about that and how to join via Zoom and everything. It's always the same link for joining Zoom. I don't have different Zoom links. It's all just one link. But FYI, so if you can't find it, the announcement, and it's that day, just click on the Zoom link at the sidebar on Canvas or something like that. It's the same Zoom link. So that'll be next Tuesday. And I will be doing the stuff, chapter lecture thing in person in here. So if you show up here in person, I will be here, but you can join via Zoom, and I will be doing the lab review in the lab. So if you want to join in person, I will be there, but you can join via Zoom is what I'm saying. So we have that option next Tuesday. All right, next Thursday, we have obviously no lecture that day, but we do have the lab midterm. So that's next Thursday. So be prepared for that. Then the Tuesday after that is our Unit 2 exam. We don't have lab on our unit exam days. So there's no lab next Tuesday. And then next Thursday, there's no lab because next Thursday is fall break. So there's no class that day. Yes, and I believe the campus is going to be closed that Thursday and Friday too. So don't even bother coming to the campus those days. Any questions about that? So this is it up to the middle of the class. Your midterm grades will be posted sometime at the end of all of this. So midterm grades come out around that time. So that will be happening around that time too. I'm hoping that they won't push us to need to post them until after the Unit 2 exam so that your grades will reflect your Unit 2 exam. For the better or for the worse, depends on you guys, right? So however you end up performing on the exam, but that'll give you a better picture of what your grade actually will be. Basically, once we get the lab midterm and the Unit 2 exam in, most people's grades don't change a lot after that. They'll change from those exams, but then after that, they don't change a lot after that. We have 2 projects that are big projects that can bump your grades some as long as you do well on the projects. And typically people do well on the projects, just saying, they're meant to be a buffer grade. So those 2 projects, again, your disease presentation project and the unknown lab, which we will go over. We'll make sure everybody's caught up on the worksheet before once we get back from fall break. We'll go over it together. We'll make sure we're caught up and that we have everything in place before we continue on with that. I will make sure everybody's caught up on that. So no worries on that, but that unknown report and the presentation are 2 big projects that together are worth 20% of your grade. So they can have an impact on your grade as well. So do well on them. People usually do well on them. You might lose 1 or 2 points here, again, just depending on how you present it, or if you do a recording and it's too scripted. Like I said, but almost everybody will get an A. Almost everybody. Now, I'm not guaranteeing you an A. So you just say, oh, everybody gets an A, I'm not going to try, then watch you get a B or a C on it. Just saying. So put the effort in and almost everybody will get an A on it on both of those projects. I'll walk you through what I'm expecting on that lab report thing. We're not anywhere near like even 10% done with it yet. So don't freak out if you haven't started that worksheet yet. I know you're like, we've done 8 weeks. It is nothing on that worksheet. So we've done a lot of lab work, yes, but it's a minimal amount on that worksheet. So if you haven't started the worksheet, don't panic. There's plenty of time for us to catch up on that because most of the work is put into it the second 8 weeks. Whenever we get back from fall break, most of the work is put in for it then. Okay, so those 2 things. So focus on those 2 things being things that can help your grade. Of course, your exams can help your grade too if you're doing well on them. So if you are changing how you're studying, you're working more on making sure that your notes are, not just copied, but now they're written in your own words, and you're following along with the lectures and engaging with them more, and you're taking time to study effectively instead of just going over Quizlets and flashcards over and over again. Then you should do well on these exams. Lab midterm and Unit 2 exam coming up quick. So keep that in mind. All right, any questions before we get going with the Kahoot? All right, today we have 3 chapters to cover. Both of these, I'll say both, all 3 Kahoots. All 3 of the Kahoots, okay, are 10 questions. We're not going to go through all 3 of the Kahoots. So if you wanted to practice the Kahoots on your own time, they are in your modules, weekly modules. This is the lecture application activity. So you can find it there, or you can scroll to the bottom of your to-do list until I enter in a grade for the participation today. Yes, lecture application activity. And that's where you can find the links to the Kahoots for each of the chapters, for any chapter ever. They're always in that. So we won't make it through all of this Kahoot. We won't make it through all of chapter 25. I will try to make it through all of chapter 10, because as I'm sure you found out making your concept maps, chapter 10 is the big daddy. So we want to make it through as much of chapter 10 as we can. So we're going to spend a little bit more time on that and discussing what my expectations are of what I would expect you guys to know about glycolysis, Krebs, and the electron transport chain. It's probably less than you think for some parts of it and more than you would think for other parts of it. For example, I don't need you to tell me that oxaloacetate combines with acetyl-CoA to make citric acid. But crap about it. I don't care about it. You're not telling me, though, if you can't tell me what the point is of the Krebs cycle, what the real goal is of it, then you're failing on that part of it. So that's what we're going to go over today. What's the point of glycolysis? What's the point of the Krebs cycle? What's the point of electron transport chain? I will say we'll spend more time going over the electron transport chain and how it works than we will over each step of glycolysis or Krebs. Okay, way more concerned about electron transport chain. All right, we ready? Let's start with chapter 9. Make sure we're signed in. If you're not, you can sign in using the code at the bottom of the screen. All right, first question for chapter 9. We'll make it through maybe 5 or 6 questions on chapter 9. The acronym CHOMPS is used to refer to the elements needed for life. What is S? See who we have on here. All right, so it's sulfur. Good enough for that. We have a lot of presence of sulfur in our amino acids. For example, methionine and cysteine are both amino acids that have sulfur in them. So in our proteins, we have quite a bit of sulfur, and they can create disulfide bonds, which is one of the important things for the structure of proteins, which we'll talk a little bit about when we get into chapter 10 and our enzymes and stuff like that. Okay, so our question for this one is Mariah here today? Mariah Brown. No, Mariah? What about Hannah? Hannah, right? Yes, Hannah. What does the P stand for in chunks? So I'll help you a little bit. Okay, well, good. That I'll take it. Yes. Okay. So I was going to give you a little preface. You didn't need it. Good job. So the P will be phosphorus, but we know it most primarily in phosphates. We know it's in our phospholipids as phosphate. We know that it's in our DNA and our RNA as phosphate. And we also know that it's in ATP as phosphate, right? So that's an important one. The N, I mean, I feel like the N is a pretty obvious one too. Is Kennedy here? I think Kennedy emailed me and said that she wasn't going to be here today. I just want to give a chance. Is Jessica here? No. All right, Barry. All right, I know you're here. Yes, so N, yes, N is nitrogen. So that's going to be that one there. All right, good job. That was an easy one. I gave you an easy question. I might have to pick on you with a harder question later. All right, so, so we know carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Okay, so easy enough. So maybe this will be a little bit more of a challenge for us if I ask a question about the nitrogen. In what macromolecules, if you can name one, there's two macromolecules that we see a lot of nitrogen in them. Can we name one of them? Anybody? So nucleic acids. All right. So Megan. It's just hard. It's not in alphabetical order when I scramble it like this. I have to search for people's names if they volunteer answers. Okay. So nitrogen. And I know the nitrogen cycle was a chapter 25 thing, but I feel like it's worth kind of going over now that we're talking about the elements. Okay. So nitrogen. We find nitrogen in our nucleic acids. This is DNA, RNA, and ATP, right? Because we know that all of these nucleic acids have nitrogenous bases. This is that A, T or U, C, G, right? Those are the nitrogenous bases. So remember that a nucleotide is a sugar, phosphate, and what are the bases? Right, nitrogenous base. So that's a nucleotide. So when we talk about A, T, C, and G, we're really talking about the bases, the nitrogenous bases. When we're talking about the genetic code, when we say A, T, C, G, we're often talking about the whole nucleotide, right? The whole nucleotide. All right. The other macromolecule, we have our nucleic acids. We also have our proteins, remember? You might not immediately think about proteins as having nitrogen in them, but if proteins are made up of amino acids, what's an amino acid? Well, it's an NH2 group, has H's, NH2. This is the NH2, linked to the alpha carbon, which has always carbon makes 4 bonds, right? That's linked to an R group, and that's linked to a carboxylic acid. This is the acid, and this is the amine. So in order to have genes, and in order to have proteins, you must have nitrogen. You must, okay? Can't live without the nitrogen. So that's the idea of why we need nitrogen. So how is it cycled in our environment? And this is less difficult than it seems, I promise. So, and I promise I want less out of you for it than you would think. N2, we find this in our atmosphere, right? In fact, most of our atmosphere is nitrogen, right? It's not oxygen. It's mostly nitrogen gas. So here's N2 in our atmosphere. We have bacteria in the soil that can convert N2 into ammonia. we call this the ammonium ion, but it's ammonia, okay? You pee out ammonia, yes? It's one of your waste products. You didn't convert atmospheric nitrogen into ammonia, though. You broke down proteins and DNA that you ate, which we talked about eating DNA, right? If you eat food, you eat DNA. It's in your food. It's in your plants that you eat and the animals that you eat. It's DNA. You eat it all the time. So nitrogen from DNA or proteins, you pee out as a waste product, the parts that you're not going to be using, okay? That is not going to be ammonification. Ammonification is going from atmosphere to ammonia, which bacteria do. So that is all you need to know about ammonification. Bacteria do it. They can convert atmospheric into gas into ammonia. We also know that ammonia is peed out by animals from waste as well. But either way, that ammonia can also be converted into nitrite, which can then be converted into nitrate. Remember ites and ates, okay? Nitrite, and this will not be the last time we see this. And this one is nitrate. We will see nitrite and nitrate again at the very end of our labs, like this lab 18 and 19, or 17 and 18, the last two. Okay, so we'll see it again. So nitrite and nitrate. All right, this whole process of going from ammonia through nitrite and then back to nitrate, this is nitrogen fixation or nitrification. Both, same thing. Nitrification or nitrogen fixation. That's all you need to know about it, okay? I don't need to know the intermediate steps. I don't need to know balanced equation. Don't care about that, but know this. And that nitrate, that is what plants can use. Here's a plant. All right, now the plant that's growing in the soil can absorb the nitrate. And then we can eat the plant and get the DNA and protein parts, and we'll use nitrogen in that form. Following? So that's the nitrogen cycle in the gist. But now you're probably like, that's not the end of it. And you're absolutely right, because going from nitrate, some bacteria go from nitrate back to nitrogen. Bacteria, again, do this. I don't want this to be, these are separate things. This process of going from nitrate back to N2 as a byproduct is known as denitrification. Okay, so we've gone through all three of them. We had, oh fine, I'll be down here. Nitrification, denitrification, and ammonification. You guys following that okay? That was in chapter 25. And that's all you need to know about the nitrogen cycle. That's it. Okay. Any questions about the nitrogen cycle? I know it's chapter 25, but we're talking about chomps and I feel like it relates. All right, so that's the nitrogen cycle. Everybody get down the notes that they want from this if you want notes, because I'm gonna erase it. Okay. My drawings, okay, good. All right, which of the following is heterotrophic? Heterotroph. Which of these is a heterotroph? One of these stands out, so it should be obvious just based on that. All righty. Okay, when we talk about nutrients and energy that are sources for organisms in general, not just bacteria, there are some major ones. Now, the book goes into a lot more categories than I expect you to know for our learning objectives. If you want to learn all of them that's in the book, don't let me stop you. But ones we have to know for our learning objectives, I kept it real basic for us. You have sources of carbon, You need carbon in order to live. We already talked about the nitrogen cycle. We could talk about the carbon cycle, but I think the carbon cycle is pretty easy, especially once we do chapter 10. Like that's the hard part of the carbon cycle. But the carbon cycle is pretty straightforward. So we won't go over the carbon cycle, but we will talk about what we term organisms based on where they get their carbon. So carbon versus energy. And I always start with the energy side. Okay. Because I always like to start with ones that use energy from the sun. We call them phototrophs. Ones that get energy from breaking down chemicals, break chemical bonds. We call them chemotrophs. So I always start there because that sets me up for remembering the other terms. I'm just being honest here, okay? Once they get their carbon from converting inorganic, we'll put an I for inorganic carbon source in the form of CO2. Now, a lot of people are still confused why carbon dioxide is inorganic. It's because the carbon isn't bound to a hydrogen. That's the rule of organic molecules. Slap yourself on the hand if you thought that having carbon meant you were organic. Somebody lied to you if they told you that. And I bet you Dr. Tim didn't tell you that. Y'all made that up in your own head somewhere. I know Dr. Tim would have told you that you have to have carbon bound to hydrogen in order for it to be an organic molecule. Carbon doesn't dictate organic. Carbon being bound to hydrogen dictates it being organic. Even whenever we have sodium bicarbonate, which this is sodium bicarbonate, this is bicarb, this is what is in baking soda, okay, normal thing. This is not organic, this is not organic, this is not organic. Because what's actually, this is where you got to understand your chemistries, right? What's actually going on there is a C and an O and some O's. This O is bound ionically to the sodium. And this O is bound to the H. The H isn't bound to the carbon. That is not an organic molecule. You have to understand the structure of the molecules in order to understand whether or not they are organic. So I get this question all the time. They're like, you're telling me carbon dioxide is inorganic. It's got carbon in it. Then you failed yourself at understanding what an organic molecule was. And I bet Dr. Singh and Dr. Kim, if you took GOB, are at you right now. If they're rolling in their graves, except they're not in their graves, but you know what I mean. Euphemism, okay? But yes, so if we can take carbon dioxide, which is inorganic, okay? Inorganic, that's what the little I means, okay? Inorganic carbon, and put it into glucose. Can we agree that glucose is organic? Yes, we know C6H12O6, that's glucose, and it's in the big old ring, and there's hydrogens coming all off of those carbons there. C6H12O6, glucose is obviously organic. That this, we call organisms that can do that autotrophs. Okay. Organisms that take glucose and then break it down. Do we use inorganic carbon and turn it into organic? No, we steal organic carbon from plants that we eat or animals that we eat. So things that already use organic carbon, we call them heterotrophs. So what kind of organism are we technically? We break chemical bonds for our energy, and we use organic carbon. So energy and carbon together, we would be chemoheterotrophs. Chemoheterotroph. Plants are photoautotrophs. Yes? Yeah. All right. All right. So all of these organisms we can agree are eukaryotes. That's on this list here. They're all eukaryotes. They all have true nucleus, right? So if we talk about bacteria, can bacteria be a photoautotroph? Cyanobacteria are. Can they be chemo heterotrophs? Yeah, of course they can. They can also be chemo autotrophs and lithotrophs, which basically break down rocks in order to get their sources of energy and carbon and stuff like that. But I don't expect you to know that in our learning objectives. I felt like that was too much. So we're keeping it on the basics here, okay? We're keeping it on the chemo, photo, and then the auto hetero. Okay, does this help us see the difference a little bit better? The lecture just kind of goes through it linearly, but sometimes breaking it down in like these categories, it helps me see it better anyway. So I hope that helps a little bit. Okay, so easy enough stuff. Yeah. Yeah.
Would Sapro be a chemotrope?
Excellent question. So yes, saprobes are chemo heterotrophs. They break down dead things. They're like a specific category of a chemo heterotroph. That makes sense, yeah? They're still breaking down organic sources of carbon. They're still breaking down chemical bonds to get energy. It's just being more specific about how they do it. That's excellent. So I'm actually going to give you points for that because that was a good question. It actually leads into the next part.
So does detritus, same?
Yes, yes. Any dead and decaying matter, the word, well, that's just a fancy word for dead and decaying matter. Detritus or detritus, however you want to say it, as long as you can recognize on the test. I say detritus, but I've heard people say detritus. I don't care, as long as you can recognize it on the exam. Okay, saprobes, like Brandon was leading into. What do saprobes do? All right, so saprobes are decomposers of dead matter and detritus. They would not be chemo autotrophs, they're chemo heterotrophs. So that's the thing to remember here. With the autotrophs, they're converting inorganic carbon dioxide. Do we have some that might do some of that? I mean, maybe they might be able to switch a little bit between. It just depends on the organism that we're talking about. But generally speaking, especially with our fungi as our decomposers, all of our fungi are going to be heterotrophs. None of them are going to be autotrophs. What else do I want to say? I don't think anything. I don't have any questions really for this stuff yet. You're fine. Okay, diffusion of water molecules through cell membrane with a concentration gradient. Concentration going from high to low, normal. Diffusion. Water is diffusing across a selectively permeable membrane. All right, so this is osmosis. So real briefly, Anastasia. Just general difference between diffusion and osmosis. So sort of, yeah, so I'll give credit for that. So we'll think about this way. Osmosis is diffusion, but is diffusion osmosis? No, right, exactly. So that's the point there. Diffusion is any movement of a molecule from high to low concentration. And osmosis is specifically diffusion of water. Okay, and then we think about life, water being the solvent of life, right? And so anything that's usually, and we're talking about biology, most of the stuff is in solution, and that solvent is water, and the solute is whatever's dissolved in water, which is coming up with the next question that a lot of people were missing on. Yes, you're fine.
The diffusion, does it require the selective permeable membrane? Is that like the difference between, besides just being water with osmosis and diffusion?
So technically when we talk, so
You have to use it without the permeable membrane?
you can absolutely have diffusion without a permeable membrane. So if I spray perfume here, like we've talked about with our antibiotics, right?
Yep.
If I spray perfume, it diffuses through the room, just like our antibiotic diffused the media. We didn't have a membrane there. But water diffusion is usually dictated by the membrane. So can you have water diffusion without a membrane? Technically. But, so anytime we have water diffusion, we would consider it to be osmosis. But in the cells, we have to think about the whole reason water is diffusing is because the other molecules can't. So if you have a cell that is full of water, let's say cells aren't full of water, right? They're full of mostly water. So here's your cell that is 95% water on the inside. And then the outside, let's say it is 50% water. Well, it's 50% something else then, right? On the outside. So it's 50%, who cares what? Something. And then on the inside, it's 5% something. Which way is the water going to flow? Out, because the somethings can't go in and out. If they could, they would too. But the membrane is keeping the somethings from moving in and out. So the water has to move in and out to equal out inside and outside. So does that make sense? So that's why, and I know you guys are really keen at saying, obviously, if it's more water on inside, water will move out. But we don't think about the fact that the whole reason that that is happening is because the somethings can't move. So don't forget that. That's where the membrane becomes important. Whether it's salt or sugar or whatever, they can't cross the membrane on their own. Water can. And so that's why the water is moving and not the other things. Okay? So keep that in mind whenever we think of isotonic, hypertonic, and hypotonic. Okay? So this would be an example of if you pack something in a salty solution. Obviously, if you're packing something straight salt, that's not going to be 50%. It's going to be like 100% something else, right? It's going to be like 100% something else and very, very little water. And that's why all the water will leave the cell, and that will cause plasmolysis. And so that's a good way of preventing those organisms from growing in that. But did you guys ever think about jams and preserves? So they don't have a lot of salt in them, do they? But they have a lot of sugar. Sugar is still a something else. So you can pack something in sugar and it has the same exact effect as packing it in salt would. And that's how jams and preserves work. We pack something in sugar. And so we're keeping that same idea of drawing the moisture out of any cells that could have made it into that solution. Make sense? Keeps the bacteria out. Okay, good. You guys are doing good with this. Don't ask me any other questions yet. I don't think so. I'm just trying to think. Active transport and facilitated diffusion. They both need energy in order for them to work. Okay, so those are going against a gradient, or it's helping things move with the gradient. So within facilitated, so I want to go about the distance. What I'm trying to get is the difference of these. Active transport and facilitated diffusion. One thing. In active transport, you need energy. Come on. You need energy. And in this case, we're going against a gradient. In facilitated diffusion, you will need to go with the gradient. But these are things that can't go across the membrane. So they're blocked by the membrane and will create a channel so that they can still move, so that there's a lot of them inside and only one or two outside, that they can then go through a channel. That's facilitated diffusion. You need a protein that helps with that, but maybe not energy, but you need energy to build the protein. So sometimes that needs energy and sometimes it doesn't, depending on if it's a gated channel or if it's just an open channel that allows you to move. So that just depends. Active transport, we're either moving things against a gradient or we want to move them faster than the gradient will allow them to move. There might be a little bit more on the outside, but diffusion isn't bringing it in fast enough and we want more of it in there. If you're eating food, for example, even if you might have enough nutrients inside the cell, diffusion might not bring it in, but you want to keep eating it up and bringing it in the cell, then you would need active transport for that. So we do need energy for that one. Let's see, Caitlin King. Is Caitlin here today? Braley. All right, Braley. What is our major energy molecule in the cell? So we have one major molecule that serves as our energy source for the cell, and we're going to talk about it a lot in chapter 10. Mm-hmm.
It has something to do with the metabolism.
It does, yes. And so it is literally the take-home message for chapter 10 for aerobic respiration. There it is. That's ATP. And so you're going to not forget that for sure once we get through, chapter 10 and we review that stuff. So chapter 10, we're basically talking about how do cells get ATP. And so if you need to do active transport, you need ATP to move things. If you're going through exocytosis, which is moving things in bulk out of the cell, exocytosis, out of the cell, then you need energy to govern that. You need to phagocytose something or endocytose something, take something into the cell, then you're going to need energy for that. If you need to make copies of your DNA, energy for that. If you need to make RNA, you need energy for that. If you need to make protein, you need energy for that. All of that is governed by ATP. Okay, so ATP is our energy. Beautiful, excellent, good. All right, let's move on to the next question. See where we are. What is the solute in salt water? Let's see if we were paying attention. One of water and salt, solute. And the other thing is, we'll ask in a second. That's going to be the question. So we know it's water, and we know it is salt. Which one is the solute? All right, so salt is the solute. All right, keep it to yourselves. We got a question coming. Esther, what would we call water? If salt is the solute, water would be the in a solution. Okay, if salt is the solute in a solution, water would be the so it's not going to just be, we talked about this back in chapter 2, and this is basic chemistry. The term for water, and we call it the blank of life. What is the term? Anybody know it? Solvent. We just said it. All right, this is where we got to get away from relying on our notes. We got to start applying the notes, right? So we're looking too much for where's the answer in the notes. I'm not asking questions necessarily directly from your notes. I'm asking us to apply our concepts, right? So it's okay, we'll give you another question. We'll come back to you. And I'll try to make it a little bit more direct with the chapter so that it's easier to find in our notes. But at the same time, we got to get away from that. We got to start applying that stuff. All right. Solute concentration outside the cell is the same as inside of the cell. So we'll ask you a question about something related to this. All right, Esther, and that should be in chapter 9. So when it's the same inside and outside, if it's the same inside and outside, what's that term? And Esther, I'll ask you about hypertonic. So I'll ask you to tell me what hypertonic is. All right, so if it's the same inside and outside, that would be isotonic. So Esther, what is hypertonic? So that would be hypotonic. So if we have a cell and we have 95% water and we have 50% water on the outside, that means 50% something else, right? In this situation, where would the water flow? We already talked about this one, right? But just again, just to reiterate, the water would flow out, right? This is a hypertonic situation, hypertonic. Why? Because hyper is up. Hyper is up, tonic situation, mixture, more salt or something. So if I'm 50% water and we're talking about salt solution, we would be how much salt? 50% salt. If we're talking about the cell is 95% water, then if it's only salt, then 5% salt. So there's more salt on the outside than there is on the inside. And so that's what makes a hypertonic solution. And again, the reason the water moves out is because the salt can't move in and out. The water can. So the water is trying to create that equilibrium. This is all diffusion. And diffusion is literally always dictated. I'll still give you points for that because we're talking about the content, so no worries there. But anyways, when we talk about diffusion, we're talking about physics. We're not talking about chemistry anymore. We're talking about physics. And you guys learned about physics in chemistry, really did or not. Do you guys remember entropy? I know you remember the term. You might not remember too much about the concept, but but it is one of the laws of thermodynamics that everything is going to tend towards a less organized state. So if I have a whole bunch of bouncy balls stacked up here, what are they going to want to do? Spread out. That's physics that dictates that, not chemistry, right? That's physics. And how much of chemistry is actually dictated by physics? All of it. How much of your body and your biochemistry in your body and how your proteins are folding and interacting with each other is actually dictated by physics? All of it. This is quantum mechanics level type stuff, but literally in chemistry, everything is dictated technically by physics. And almost none of you guys are going to have to take physics, which is a sad thing because it's the real answer to what is going on with everything. Why atoms work the way they do, why we have a nucleus even, why we have electrons and why they behave the way they do around. That's all physics. That's not chemistry, that's physics. And so is diffusion. So if I have a whole stacked up bouncy ball tower here and I hold it together with a net because the bouncy balls don't want to stay stacked in a tower because physics says they want to spread out. That would be true whether there's gravity or not. If I have, even without any gravity at all, in a complete vacuum of space with no gravity or anything else holding them together, if I have a whole stacked up bouncy ball thing and I cut the thing open, the bouncy balls are still going to spread out in space over time. That is diffusion. That's entropy. That's a law of thermodynamics, not a theory, a law. And so everything's going to want to diffuse. And so if the salt wants to equal out inside and outside the cell, and the water wants to equal out inside and outside of the cell because physics says that they want to, then the only thing that can move is the water. So the cell gives up as much water as it can to try to make the outside and inside equal. Obviously, if you're one cell in a big old cup of salt, giving up all your water isn't going to do much, but it's going to try anyways because physics tells it to. And so that's what's really happening with that situation, is trying to equal out everything. And it's not consciously doing that. Physics just makes it do that. Okay, so that's what's really happening. So in a hypotonic situation, if your cell is 95% water, here's our cell, and we put it in 100% water outside, this is a hypotonic situation. Hypo, low. Right? And so now where would the water go? Inside, exactly. Because now we're trying to balance out what's inside and outside. They're always trying to balance out, even if they can't actually achieve it, they'll try. So that's the idea of what's happening in there. Right, so we have an activity that's associated with this. I'm going to go ahead and say we are done with this Kahoot. Does anybody have any questions about anything left in chapter 9? Chapter 9 continued to talk about symbiosis as well as bacterial growth curves. Did anybody have any questions about any of that stuff? Yes.
I didn't understand what, it was one, categorized organisms based on their type of symbiosis. I wasn't sure about what you were asking for.
So don't think too much about it when I say that in the learning objective. What I'm saying is, for example, I'll give you an example, okay? Virus. So if we talk about symbiosis, so without Brandon answering the question, Adolfo, we'll say, okay, you're in our next victim. Can you name one type of symbiosis? There's 3. There's only 3. Can you name one of them? And we'll go from that. We've talked about one of them continuously throughout the semester. We'll let Adolfo cook on this. Yeah. Yeah, probably did.
Well, I just wanted to make
Yeah, don't overthink it. Yes.
Would it be parasitic?
Sure. Beautiful. Okay, so that's the exact one I was thinking of. Parasitism. 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 Commensalism. And mutualism. In parasitism, always in symbiosis, is 2 organisms that one of them depends on the relationship for its survival. This is not an optional relationship. You and your dog, that is not a symbiotic relationship. Your dog can leave you and go find food in the wild. If your dog died, literally, I don't mean like it happened to die. I mean like it always died if it didn't have the relationship with you. That would be symbiosis. So for example, leeches. So if I talk to you guys about leeches, leeches have to feed on other organisms to live, right? Viruses have to grow inside of your cells to live, and they use all of your cell's resources to do that. So those are parasites. That is a symbiotic relationship. Symbiosis, we are too used to. I don't know who taught you that a symbiosis was a good relationship, because that's not what symbiosis means. Symbiosis means two organisms depend on the survival, one of them for livelihood. That can be good or bad. If it is a bad situation where one is harmed, that will be the host is harmed, and the parasite benefits. Like a leech, like a virus, like any bacterial infection that you have ever had that's caused disease in your body, okay? And the parasite benefits. That's parasitism. In commensalism, one benefits, the other has no effect whatsoever. It's not harmed or helped. It's just existing. So leeches cause you harm because they're taking blood out of you. But if you're a sloth, for example, and you have algae that lives on their back. Now we could argue that maybe the sloth is benefiting from the algae on their back, but I feel like that's a reach. But the algae that grows on their back, sloth climbs up in the tree, the algae gets sunlight. Is the sloth harmed or helped? Sloth doesn't care either way. Sure, I mean, we could try to, yeah, we could try to argue it a little bit, but generally speaking, in the overall big picture, that would be a commensal relationship, okay? Mutualism, both benefit. We should be able to give an example of a mutualistic relationship. This would be, for example, the bacteria in our gut. The bacteria in our gut are breaking down things like food for us, and their byproducts of breaking down that food. Now we can digest the nutrients. They are getting, housing and food for themselves as well. So that would be mutualistic. We're both benefiting from that. There's definitely goods on both sides of that. They're helping train our immune system and stuff too. So that's also a benefit that we could say that we're getting from them. And the list really goes on and on with that sort of relationship. So we see the differences between these symbiotic relationships. We're used to thinking mutualism when we think symbiotic, but we got to get away from that. Parasitism is symbiotic as well. Be good? Yes.
So it is not about like living together.
Well, they have to live together. Like I would say that there's some aspect of their lives that are together, right? So when we talk about viruses, for example, or bacteria that infect us, during that time where they're affecting us negatively, we're living together, right? But you don't have to permanently live together.
So it's not a positive. Is that what you're saying? That this is like,
Mutualism is,
but not the.
not parasitism. But
Yeah.
symbiosis overall,
Yeah.
symbiosis, right. So symbiosis overall, I would say is just saying that an organism relies on another organism to live. They, like you said, they don't always live together, but there has to be some transiency of togetherness,
Okay.
like a
I was
leech
gonna-
or a tick. Does that make sense?
Yes.
Outside of parasitism, in commensalism and mutualism, since no one's being harmed, would they both be considered symbiotes?
Yeah. I mean, yeah, and we could consider parasitism to be symbiotic as well. I mean, symbiotic, they're symbiotes too. It's just, there's a negative, they're more specifically parasites, right? Yeah, so yeah, just depends on whether you're having harm or not, if we can define you as a parasite. All right, we're tracking with that and how those are categorized. They're like under the tree of symbiosis. Yes. All right, that's a good question. Any questions about I feel like the growth curve is pretty straightforward. Lag phase, then log phase. Log is logarithmic. So it's that doubling exponential is also the name of that phase. Log or exponential phase. So lag phase is just you got into a new environment. You're just taking score of what your new environment is about. We aren't growing right immediately. We're not growing immediately. We're saying, oh, there's nutrients here and there's water. Okay, now we can think about starting to grow. Once we actually start doubling our numbers, that's the exponential phase. Then we get to the stationary phase. This means that death and growth is the same about, and we slowed our growth a little bit. stationary phase. And then obviously death is where death numbers start outnumbering growth numbers. We start declining. That's the growth curve. It's pretty straightforward. Any questions about it? So that'll make more sense when we get into chapter 13. Yes, chapter 13, where it starts talking about disease and disease progression, because the growth curve, lag, log, stationary, death. Log, also known as exponential. Stationary and death. Also follows the progression of disease. We'll get to it in chapter 13, which you'll start learning about the prodrome. You'll start learning about, so incubation period, the prodrome, the acute phase, and convalescence. They directly align with all of this stuff. So you know that whenever you get exposed to somebody that has COVID or the flu or a cold, you don't get sick the second that you are interacting with them, right? You have a period of time where that virus gets into your body and it's readjusting to its situation and it's starting to multiply. And then later you start showing initial symptoms. You get that scratch in your throat, for example. You start feeling you wake up in the morning, you got scratch in your throat and you're tired. You're not full blown sick, but you know what's coming, right? So that period of time from whenever I, walk near Barry who's coughing all day and I feel fine for a day or two, that's the incubation period. And that scratch in the throat a couple days later, that's what we call the prodrome, the initial signs and symptoms of a disease. Then you start getting real sick, that's the acute phase of the disease. When you start feeling better, that's convalescence. So you've heard some of these terms, but they directly relate to the growth curve of our organisms as well. It tracks, right? And so we'll talk about that more in chapter 13. Yes.
Is quorum sensing going on throughout the entire vaccine?
I'm glad you're bringing that up because they brought it up in my other class and I forgot about it.
And so so during the phase, I mean, it sounded like what I picked up was that it dealt with, food and the environment and stuff. But it said in the quorum sensing that it was releasing chemicals.
Yes.
And so do those chemicals, I mean, does that cause cells to die or is it just because of the environment's going south?
So it could be either, honestly. So that's why I just say vaguely chemicals.
Okay.
So think about it this way. If we all lived in a building together, right? And we literally lived in a building together and you smell smoke, that's a chemical signal that's telling you you should leave the building. It's the same exact thing. So it's general stuff like that. But also, if you're walking in that building and you're going up to your floor and you see blood dripping down the steps, then you'd be like, maybe I'm gonna go out. We need to go back to, I always want to say Wawa because that's where I used to go. I don't know why I always think that, but they don't have Wawas here. On cue. I'm gonna go to on cue for a little bit and call the police because this is wild. So the bacteria have that same sort of a thing. I'll get to your question in a second. They will say, I smell smoke, or they will say, I see blood on the steps. But to them, it's like, I see parts of bacteria. Like DNA. I sensed that DNA was nearby. DNA is supposed to be inside of a cell. Something's going wrong in our biofilm. Stuff like that. Or I can sense poisons or toxins that are bad to me. I'm going to leave the biofilm. And they can also send out chemical signals of their own, just saying, I'm fleeing. And then the other bacteria, it's like leaving a note or texting their buddies. I saw blood on the steps. I'm getting out of here. And they get messages like that too. All of that is quorum sensing. So those are negative quorum sensing. We can also send out texts to our neighbors in the building saying, hey, they left cookies in the lobby. And then we might all go down and get cookies. That's also going to be quorum sensing. So it's very similar to the chemotaxis and the phototaxis with the motility, but in that biofilm sense. So it doesn't really apply necessarily to the motility, but just like communicating with the other organisms, letting them know. It's not necessarily conscious. They're just chemical responses to the environment. Yes.
Well, you pretty much answered it, but I was gonna ask, does that have to do with algae and how they communicate with each other?
Yes, it's the same idea. And so when we think about quorum sensing in our textbook, we mostly think about it in terms of bacteria, but quorum sensing does apply to things like protozoa and protista, which algae are protista, protozoa, protista, molds, and bacteria all can kind of communicate with each other on that. And so it is the same thing. And what's your name? Okay. Can't ever remember your name for some reason. When I see your name, I remember you. You know what I mean? Anyways. All right. let's call that for chapter 9. Any other questions for chapter 9? Yes.
Understand what is that, abbreviation, it's like CRISP-9 or something like that?
CRISPR Cas9, that was chapter 8. So
Okay.
no
sorry,
worries.
But I saw that for some reason, I don't know what I was looking at, but anyways, obviously that was chapter 8. I really didn't understand that, but we can talk about that later.
It's fine. We'll go over it a little bit in lab because
Oh, okay.
it relates to some of the stuff we'll talk about in lab. Yes. So we can talk about it then. All right. So we'll call that for chapter 9. We'll do a question or two for chapter 10. This is not the right thing. Cool. Here's our little podium so you guys can feel good about it yourselves for a little bit. You know, chapter 10's coming, so take a breath. We're gonna do 25. Couple questions, 25. I know, chapter 10 is a big one. So now you guys see what I mean with chapter 6 and 10. They're biggies. That's why most of the test is that. I mean, diffusion and stuff, we've heard about it before. It should just mostly be refresher, that stuff. But that's why a lot of it's gonna be 10. Hopefully what I have to say about 10 will help clear it up. Okay, so I'm gonna just pull up chapter 25. We're just gonna continue. And if you need to re-sign in, that'll be okay. Double check that you're signed in. And if you're not, you can still join using the code at the bottom. All right, chapter 25. We're gonna do chapter 25 and then we're gonna go to the activities for 9 and 25. We're gonna just walk through them real quick. Blank, what is more potent as a greenhouse gas than CO2? As a greenhouse gas. I don't mean more in our atmosphere. I mean more as a greenhouse gas. All right, so our choices here were methane, carbon monoxide, nitrogen, or hydrogen. And so it happens to be methane, and this was covered in chapter 25. This got my video flagged because I was talking about global warming, and it's a touchy subject. And I guess for some people that got flagged, not everybody, but just because I'm your setting on YouTube. so again, I am not saying that we caused all of global warming, but I am saying there are things that we do that contribute it. And that's why I got flagged, because it's like, oh, you're talking about global warming and the cause of it, and we don't know for sure. And so that's fine. Are we contributing some to the greenhouse gases? It's ridiculous to say that we're not. I mean, it's quite ridiculous to say that you're not. You exhaling contributes to the greenhouse gases. Of course, we're contributing. Is all of our gases causing global warming? I can't prove that. Nobody can, okay? Probably, but I can't prove it, okay? All right. The global average temperature is rising, and it is quite significant if you've actually seen the charts for it. So that's arguable. Global warming is happening. Whether or not we caused it, I'm not going to have that argument. But there are gases that we make and that our industries make that can contribute to that global warming. One of them is carbon dioxide, and we know that. We know carbon dioxide is a greenhouse gas. It acts like a blanket in our atmosphere, basically keeping the heat of the sun trapped inside of the planet. The greenhouse creates a greenhouse effect. So carbon dioxide does that, and so does methane. Okay, so methane. So what's the primary producer of methane here in this country? Cattle, yeah, cow farming. Cattle in general, whether it's cows, sheep, goats, whatever, but here in America, it's mostly cows. So agriculture also has a big impact on our climate, not just the cattle industry. This is something that I feel like a lot of people, even vegetarians, don't think about, which is farming in general. And I don't mean the tractors. What I mean is having that many plants that produce fruits, living in close proximity to one another on large swaths of lands, produces a lot of heat. And that has literally changed the climate of local areas, even in the United States. But if you went back 20,000 years and looked at Kansas back then versus Kansas now, I don't just mean cornfields would be there. I mean, the actual climate around the cornfields are different. It is different. It is measurable. You can see it on satellite. So that's pretty insane too. So agriculture industry, whether it is cattle or agriculture like vegetation, has drastically changed our local climates and possibly our global climates. Okay? But methane specifically, methane produced by cattle. Now the bacteria, what we call methanogens, bacteria like and archaea, produce methane in the gut of the cow, and then they fart. You fart too, okay? You make methane too, but we got a lot of cattle that are living on our lands that are farting a lot more because they're breaking down different kinds of foods, plants all the time, and they need the bacteria in their gut to help with that breakdown, and then they produce a lot of methane. And this has produced a significant amount of methane, potentially leading to global warming. Make sense? Okay. Good. Right. Yes, question.
And when you drive by a wheat field, doesn't matter if it's 120 degrees out, like the temperature drops considerably.
That has to do with how the wind moves through the wheat fields, but yes, you notice changes in the atmosphere from agriculture, whether it's increase or decrease. You can't tell me it's not changing the local temperatures and stuff like that. So they can create heat. They can also affect how the wind is blowing through those areas where we normally would have maybe other vegetation in those areas. I mean, there's a whole thing about it. If you're interested in it, please, look it up. But all right, plastic, obviously islands in the ocean. We have one the size of Texas that is in the Pacific Ocean. It's literally an island of plastic trash in the Pacific Ocean, the size of Texas. And that was when this book was written, which was in like 2022. So it's probably bigger now, but you can imagine how much light that's blocking out, and all of the organisms beneath that light that would have relied on the light, like plankton and stuff like that, no longer can survive at the level that they need, and the things that feed on that, and the things that feed on that, and so on and so forth. We're talking, hundreds of miles of light blocked out of there. So that is crazy to think about. So this one's true. Being just aware of our environment. All right, we'll do during photosynthesis, carbon. What's happening with carbon in photosynthesis? Obviously, this relates a little bit to chapter 10 as well. And we'll come a little bit full circle with this, get a little more specific with it. All right, so it's converted by organisms from a gas, carbon dioxide, into carbohydrates. Really briefly, before we get into chapter 10, where we will talk about photosynthesis. In photosynthesis, light is used with carbon dioxide. This is very general, right? If you looked at chapter 10, then you know I'm really watering it down. To convert carbon dioxide into C6H12O6, which is glucose. Whenever we talk about cellular respiration, specifically aerobic respiration, you think respiration is breathing, and you're not wrong. Okay, you're not wrong, but that is very basic level of respiration. The whole reason you breathe in and out is because of glycolysis, Krebs, and electron transport chain. That's the only reason you breathe in and out. Literally the only reason you breathe in and out. So don't disconnect breathing in and out anatomy-wise don't disconnect it from glycolysis, Krebs, and electron transport chain, because they are the same thing. Okay, so what's really happening then, what do we do with glucose? We convert it basically into waste product of carbon dioxide, and we make some ATP from that, right? That's our gist of what we're getting. So CO2 is our waste, and we get some water as waste too. We'll talk about how that's all done in a little bit, I promise. We'll walk through it in a little bit to get there. But we breathe out carbon dioxide. Now you notice I took glucose, so this was, plants do this part, right? We know plants do this. We know all of our photosynthetic organisms do that. But do they also do this? Yes, they do, my folks. Yes, they do. Interesting. In our picture here, we know carbon dioxide photosynthesis is done by plants. Plants use the carbon dioxide to make the glucose. What did we say all eukaryotes have? This energy-making organelle? Mitochondria. All eukaryotes have mitochondria. The only function of mitochondria The only function is the electron transport chain. That's it. And that, if you have a mitochondria, is always aerobic, so it needs oxygen. It uses oxygen to break, we've already broken down glucose in glycolysis and Krebs. That happens in the cytoplasm. The electron transport chain is what happens in your mitochondria. And that is where we're going to be using oxygen, breaking down oxygen, making water as a byproduct to make ATP, right? So, hey, if all eukaryotes have mitochondria, including plants, plants also use oxygen when they make ATP, when they break down their glucose. So they're making glucose just to break it down in their mitochondria and everything to make ATP. We're going to go through that in aerobic respiration, but that's all that mitochondria do. So if plants have mitochondria, they are also going through aerobic respiration. They're also using oxygen. Yes, I know they make oxygen. They make more oxygen than they use, but they also need to use oxygen. You tracking? Most people, I don't know why that gets lost, but if they got mitochondria, mitochondria only does that. Yes.
I guess I was thinking plants had the chloroplast. They do.
They have both, all eukaryotes, period. Okay? And all mitochondria always do that. So they have chloroplast and mitochondria. Yes. So chloroplast is where the light reactions occur. Remember this part? That was in chapter 10. So we'll go over it all in a little bit, but keep that in mind. We're going to come back to it. Okay. We're going to do our activities really quickly. I do not want to spend much more time on this. So I'm just going to, no, cancel. Okay, let's quit this. And now, here's our little podium. Clear my drawings, and we're going to get chapter 10. So I want to spend the rest of the time on chapter 10. This is still less time than I meant to spend on it. Today's lab, this is my other warning. Today's lab is going to be a short lab. So lecture will leak into lab time a little bit because I really want to cover chapter 10. Okay, but we will still be done with lab. It should be early. Okay. Definitely won't go over time. Not lab, not even if we carry over here. And I'll still give you a 10-minute break in between. I promise. Okay. So let's pull up a new one. Let's pull up chapter 9 or chapter 10 so that I can have it ready. Chapter 10. So some of us won't have to sign back in. All right. Our activity for chapter 9. What is the difference between diffusion and osmosis? We already went over that one. Remember, osmosis is a type of diffusion. It's just diffusion of water. Saline is often administered to patients to keep them hydrated. Do you think saline is hypotonic, hypertonic, or isotonic compared to a patient's blood? Defend your answer. Tawny, what do you think? Isotonic, perfect. So we will say this. We want saline to be isotonic to what a healthy human being's blood would be. Your blood might be hypertonic to that, but we want to bring it back to isotonicity. And it won't be, even if you're severely dehydrated, it won't be drastically different. It might just be a little bit. So in order to hydrate you, we want to be sure that there is some salt there. Because, and that's the next question, what would happen to a patient if we gave them pure water instead of 0.9% sodium chloride? So so we'll see. Mikey, because Mikey's our next victim. No offense. What do you think would happen if we put pure water into a patient? So this is, I'll put it in perspective. Our cells are, we'll start here. I'll walk you through this. If our cells, here's your red blood cells in your bloodstream, okay? Are your red blood cells pure water inside? No, right? So let's say that they are 95% water, 5% something else. If I put 100% water in your IV, what's going to happen? The water will go, well, which way is it going to go? Into the cell or out of the cell? Into the cell. Perfect. Okay, so that's what we're getting at. And then what would happen to the cells as a result of that? They'll swell and possibly burst. Beautiful. Yes, exactly. So if I give you a little bit of water in your IV, just a drop or two, your body will probably compensate for it and we'll be okay. But if I give you a whole bolus of water, if I give you like an IV bag worth of water, your cells, your red blood cells first, because that's what's going to be in your bloodstream. And potentially the cells lining your blood vessels will swell up with that water. And red blood cells could burst. Your body's going to start recognizing that red blood cells are bursting. And it's going to be like setting alarm bells off immediately. Why are red blood cells bursting? That could cause you to go into shock very easily, which is just an immediate response. If you have red blood cells bursting throughout your body sends signals saying shock time. And that lowers your blood pressure and increases respiration and things like that, but can lead to death very quickly if it is not managed well, right? So shock is there to help preserve the brain function, keep your heart pumping and stuff like that. But if it goes on for too long, you know, we're trying to allocate resources to keep the blood pumping from your heart, but you actually lose circulation to extremities and even to your brain if it's left for going for too long. So that can lead to death. Also, those ruptured red blood cells now aren't carrying oxygen or carbon dioxide for you, and they're being cycled through your spleen, which gets rid of that stuff. So that's putting a lot of pressure on your spleen, and that can lead to issues with your spleen as well. So we've got a serious situation that could lead to death very quickly and might even need blood transfusion and things like that to recover from. I mean, could you kill somebody this way? Technically. But it would require more than just a shot of water. You would need quite a lot of water because your body is meant to compensate for that. So your kidneys do most of that function for us, right? Your kidneys are regulating your electrolyte balances in your blood, as well as removing any of your waste and things like that. So the first thing that might be affected might actually be your kidney, because your kidney is really trying to compensate for how much water is in your blood and trying to keep things isotonic. If you put straight water in there, you're going to be damaging your red blood cells first. Then your kidneys trying to compensate and balance everything out with maintaining all that. So that gets into a lot of physio and anatomy stuff, but it relates. Yes.
You talk about isotonic, hypertonic, or what perspective? Is that from the perspective of the solution, the cell,
So it will always be in the question. So hypertonic isn't itself a term that tells you whether it's in the cell or outside of the cell. I will tell you in the question what it is. So in this question, it's saying saline. Do you think saline is hypo, hyper, or iso compared to blood? And so here it's iso because the saline is the same as the blood. Now, if I were to say we're giving your patient a 20% saline solution, is that saline solution iso, hyper, or hypo? Compared to blood, you would say it's hyper, right? Because it's more stuff in it. We tracking? So it's always in relation to something else. And I will always say it in the question. So if I were to ask about the blood compared to the saline, if we had, let me write it out so that so we can ask this question, okay? If we had a 20% NaCl solution, and then we had blood, and I were to ask you, is the blood iso, hyper, or hypo compared to the saline? You should know it. Is your blood 20%?
No.
Right. Your blood is like 95% water, right? So only 5%, it doesn't matter what else. Hypo. Yes. We know blood is like 95% water. We can all kind of agree on that. Yes, we know it's mostly water, at least. It ain't going to be 80%. It's going to be a lot higher. So, I mean, I could make it more drastic than that. And generally on a test question, if you come to a test question, I'm asking you about blood and you're like, what is blood though? Is it 80? Like, I don't know what the exact ratio is. And just ask me. That's a fair question on that sort of stuff, right? If you had a question like that on a test.
It's mostly water.
It's mostly water. And we know that too. So don't overthink that stuff either. Yes. If it's not obvious, it's probably because you're overthinking it. I mean, no offense, right? Okay, so are we good? Are we tracking that? Next is our chapter 25 questions. It talks about COVID in water. So one of the things that we didn't get to on our Kahoot was water treatment. And so it does walk you through water treatment. I feel like it was very straightforward on how it is done. Water treatment. We have how your tap water is treated versus wastewater treatment, and they are different things. So your water sources come from places like Oberholzer and Lake Hefner, and they get treated in a treatment plant before they come out to your tap. Okay, so there's a whole system and you don't think about it, but what's really happening in, those aren't Lake Hefner and Lake Oberholser aren't just lakes to hold the water. They are sedimentation lakes. And so it's literally meant for the water to have stuff settle out of it. That's the first step in our water treatment, letting stuff settle out of the water. Then we take the top layers of that water and put it through our filtration process before it comes out of your tap at home. So now whatever treatment is going on at the water plant, we're going to be removing solids, removing anything, filtering big particles, filtering smaller particles, and then we will go through that UV treatment and then chlorine treatment as well. We add chlorine and fluoride back to your water, whatever. So cool enough, we kind of have an idea of that. You do understand that your drinking water that you get out of your tap and that you shower with at home traveled miles through pipes underground and that those are not clean, yes? Don't, yes. I have an RO system at my house for a reason, okay? So I live in Bethany. Bethany has the worst water in the entire state of Oklahoma, period. Like, this is not an opinion, this is facts. It has been tested multiple times. It is borderline illegal how bad the water is there. We're talking Flint, Michigan level bad water in Bethany, Oklahoma. So if you or a loved one lives in Bethany, make sure they are not drinking tap water. They have boil controls where like they'll tell you like you need to boil your water. It's not safe to drink it like once every couple of years, easy, because there's E. coli or something in their water because it's terrible. Bethany water is bad. If you have well water, don't sit there sitting on your booty thinking like, oh, we have well water. It's not, who's testing your well? Because the city typically isn't testing it for you. You're supposed to test your own well water, and it's probably not clean either. So well water and Bethany water, bad. If you're living anywhere else in Oklahoma, you're probably okay. But you got to think about that water is traveling through all these pipes. And when's the last time them pipes was cleaned? Never, probably. Okay, so that's what I think about when I drink my drinking water. If you're showering in it, that's one thing. But if you're drinking it, that's another thing. I am not advocating that you don't drink tap water. I still drink from water fountains and tap water and stuff too, okay? I'm just saying be mindful about it. Be aware of it. And if you start feeling sick, have you considered that it might be your water? So I live in Bethany. I have an RO system for my whole house, and I have another filtration system on my tap where I get my water from that I drink. Because I live in a house that was built in the 60s. All my pipes are really, they're old iron, like cast iron pipes or whatever. They're crap. And so they're corroding and stuff like that, and I don't need to be drinking that. Now, we talked about isotonic, hypertonic, and hypotonic, right? So if I have an RO system in my house, if it's pure RO water, then that is pure water. It is 100% water. There is no vitamins or minerals in that water at first. I have to add back in what they call water softener, which is the minerals and the things like that. So that when I drink my water, it is not pure water. Because if I drink pure water, that's not good for my cells. We just agreed on that. 100% water is bad for your cells, whether you drink it or bathe in it. You have to have minerals in your water.
So how does that connect to having hard water?
So hard water just means you have too many minerals in it. Generally speaking, I'm not saying hard water is good for you, right? There's a lot of studies that are being done on this and like the overall long-term effects of how good your water is and stuff like that on the body. And I don't have a lot to say about it. Another student asked me about like hormones, for example. So women on birth control, there's a lot of women that take birth control and you pee out estrogen. And then this goes through our water cycling process. We release that estrogen back into the environment. Now, before we take the water from the environment and put it into the tap, it goes through enough treatment that what you get from your tap isn't going to have appreciable amounts of estrogen in it. But the water that we release back into lakes, rivers, and streams after we treat wastewater does have estrogen and stuff in it. And it might not be appreciable to you with your minimal exposure with it, but the fish that live in the lakes and stuff like that, there has been shown evidence that the hormones and the things like that that's in our waste products are affecting. And those aren't filtered out in wastewater treatment. Releasing back into the environment. Treating it before it comes to your house, tap water treatment, it does often filter that stuff out. But back into the environment, it's affecting our environment. It's pretty crazy. So I don't know everything about that stuff, but just think about it, right? Think about it and how it's affecting our environment. So that gets back to chapter 25 in our One Health thing. The whole health of the whole world, not the health of human beings, the health of our entire world and how that affects us long-term too. So if we're affecting the fish and then we go and eat the fish, and then now those fish aren't the fish that we used to eat, they're different fish now. And how much is that going to affect us? And I don't know, I don't have an answer to that, right? But think about it. So anyways, so back to water treatment, chapter 25, we to think about COVID. When COVID started happening, COVID is released through our feces and our urine in appreciable numbers. It's significant shedding of the virus in your urine and your feces. COVID is not a respiratory virus. It is not. It gets in that way, but that's like saying that the measles is a respiratory virus. Measles is transmitted respiratory too, but we know that it mostly causes skin and neurological effects. So it might be transmitted respiratory, but it actually infects almost every single cell in your body. COVID does. So almost every cell in your body, including your digestive tract and other in your heart and things like this, right? We talked a little bit about that, but one of the things we had to be aware of when COVID started being an issue is our water treatment plants weren't filtering out the virus. Viruses are much smaller than bacteria, and this is where the second question is in, okay? So the first question asks us, why would it be a public health concern with COVID if it's admitted in the feces and the urine? Because wastewater treatment, we might treat wastewater and release it back into the environment, but it's not removing any viruses. It's removing bacteria. And so now that's back into the environment, it cycles back in the environment, animals can get the disease, and then people can get the disease as well whenever we start cycling back in the other way. So that's a big issue. The second question is, would fecal coliform testing be helpful for identifying the risk for the presence of SARS-CoV-2? So fecal coliform testing is using indicator bacteria. So we know you have normal E. coli. There's a lot of different kinds of E. coli, right? We're learning about that. If you have normal E. coli in your poop, if you have Klebsiella, if you have Enterobacter, there's a ton of different bacteria that's normal in your poop. If we see that, those indicator bacteria, normal things that don't usually cause disease, if they're making it through the filtration process, then Salmonella, Shigella, bad E. coli, cholera can also make it through the filtration process. So the coliforms are indicator bacteria that are normal fecal bacteria, that if they make it through, then it's safe to assume the pathogens could also make it through, okay? So if we're checking for the presence of E. coli and Enterobacter, normal coliform indicator bacteria, if we're checking for that, and let's say it's 0, we treat our wastewater and there's no coliforms, would that mean that there's no COVID in there? Beautiful, no. Why? Because viruses are much smaller and they're not filtered and they're not removed the same way that the bacteria are. They're too small. This was a huge concern with polio. You don't know because we weren't really alive for polio, but polio is only found in people. It's not transmitted within animals. There's no animal reservoir for polio. It's only a human virus. And it's such a tiny virus that it was escaping water treatment. It's so tiny, it's smaller even than COVID is, but we had to learn how to filter out polio. We don't worry about it anymore because people in America don't have polio. I had a student even ask me once in my last class, they're like, well, couldn't people bring polio back over here? What person with polio is trying to travel and visit the whole world? Have you seen what people with polio look like? They ain't going anywhere. They're bedridden, they're paralyzed. They're not going around the world visiting America. They're not bringing polio back here. And so if they were sickly like that, they are going to get caught at customs too. So we're going to be like, maybe you don't come here. Because we don't worry about polio anymore because it's been eradicated from the US. There is polio in Afghanistan and Pakistan. That's the only place in the whole world where polio exists anymore. And it's just because vaccine efforts have been poor. I have a student in my Monday morning class who is from Pakistan, and she was telling me about this, about how people just have a poor view of vaccination in general, not unlike how our political climate is steering things right now. I think we can agree. So they have a much worse view of that and medicine in general because it is a third world country. especially in the rural areas. It's very third world. And so their exposure and understanding of the benefits of vaccine is very poor. And so they have not been vaccinated and the efforts have been rough. She went back home, she said last year, and there was a girl who was having to crawl around in the streets. And she was thinking about like, why is this girl crawling around? That's weird. And now it occurred to her that she was like, I'm pretty sure that person had polio. Because they don't vaccinate well for it. And if they give the oral drops for polio there, we don't do that here in America. But anyways, so they're trying to eradicate it from the world. And I think we will see it eradicated within the next 10 years, I'm hoping, because it is a debilitating disease. Once you've had polio, many people are affected for the rest of their life from having had it. And if you've seen the images of people in iron lungs, it is no joke. The last person that was in an iron lung died last year, I think. Yeah, for like 78 years, this person was on an iron lung. And they got like 2 PhDs and like all that makes me feel like I'm not doing enough. Paralyzed for 78 years or something in an iron lung. And they accomplished a lot more than I have with all 4 of my working limbs.
They were just chilling like, they had nothing better to do.
Yeah, I guess.
We could
Yeah.
all day long.
Yeah. It's wild. Yes, yes. Yes. Okay, good.
Yeah, I think still.
Okay, I just saw your notes on here. Sorry, that was my bad. Okay, so moving on, we're gonna move on to chapter 10. Make sure you're signed in really quick. Like I said, we're gonna carry over a little bit, but I'll give you guys a 10-minute break before we meet in lab. Okay. The lab will be shorter. For sure. And I will walk you through aerobic respiration when we get to it. I will walk you through the enzymes. Okay, and then if you guys have any specific questions about that stuff, hopefully we can assess that too. Alrighty. All right, we're gonna go ahead and get going. Remember, you can always sign in using the number at the bottom of the screen. This number will appear at the bottom in a second. All right, chapter 10, enzymes and metabolism. Which term refers to all of the chemical reactions that occur in the cell? Mariah never showed up, right? Mariah. Did Kennedy ever join us? I don't know who is online here. I know Cora is one of the people, I think. All right, I think it's Victoria. All right, metabolism is the term for that. So, Kennedy's still not here, right? Did Jessica Morales show up? All right, Caitlin King. And I think, again, I think it is Victoria and Cora that's on here. My Cherise. Yeah. Okay. All right. No, let's not do that. Let's do catabolism. What's catabolism? That's your one. Right. So generally speaking, with our enzyme world, we have all of our stuff, metabolism. Within metabolism, we have breaking things down, and that gives energy. This is catabolism. Then building things uses energy. Obviously, if you're building something big, if you were building your own log cabin, that's going to cost you a lot of energy personally to build that, right? It's the same thing for our cells. That is anabolism and anabolic steroids. Anabolic steroids are for building muscle, right? So that's the idea, building. It needs energy to build. Anytime we are breaking something down to give energy, and use that energy that we made from breaking something down to build it, that is where amphibolism comes in. Pairing the two together. If I'm breaking something down and getting energy from breaking it down, and now I'm using that energy to build something else, that's amphibolism. For example, you ate food, you broke down the food to make ATP. Now you're using that ATP to replicate cells to repair a cut that you got. So replicating your DNA, making new proteins and things like that, using energy that you got from the food that you broke down. That's amphibolism. Okay. We tracking? Good. All right, next question. Go. All right, question 2, anabolism. I know I just erased it. Let's see if we can recall it. All right, so anabolism, building molecules by using that energy that we got from catabolism. We're going to move on to the next question since we already covered that. All right, question 3, enzymes are an example of what type of macromolecule? Most of our enzymes are going to be proteins. For this class, we will consider them to be exclusively proteins. I'm not going to talk about any other kind of enzyme. They're all going to be protein based. Now, coming back to that idea of our different types of enzymes, we have 2 types of enzymes. I know that you're like, there's 6 categories. I'm not talking about that. I'm talking about 2 major types of enzymes. You are either simple or you are conjugated. We're going to spend more time on conjugated because obviously they're more complicated. Okay. Simple enzymes are just protein. They might be multiple levels of protein, but they're just protein. Okay. So they could be one polypeptide chain folded like this, mixed with another one folded like that, and then it works, but it's just protein. And remember, if we have multiple polypeptide chains, that would be our quaternary level of protein. There. How are our enzymes working? Remember that at our primary level, we were just amino acids linked together. You guys remember this? One amino acid, another amino acid, another amino acid. This is from chapter 2. This is where I said that we don't ever really get away from unit 1. Secondary level. Those amino acids have some sort of R group on them that might attract other R groups on the one next to them, or repel other R groups on the one next to them, or whatever. And then this leads to alpha helices, which again, this is just amino acids that are repelling or attracted to each other. Alpha helix, or beta pleated sheets, where we have amino acids linked next to each other, etc., going one way, and then they curve around and they go this way, and they curve around and they go this way. This is a beta pleated sheet. And this is because those amino acids are attracting each other like this. These are hydrogen bonds, for example, or ionic bonds. H or ionic bonds. All right. I know we went over this before, but are we getting a better perspective on it now from what we have covered? Okay. Tertiary This is where we take all those alpha helices and beta pleated sheets, and now they are interacting on top of one another. And then maybe there's another alpha helix and there's some more beta pleated sheets and then some more alpha helices and whatever. However, these are interacting, we might create more hydrogen bonds across bigger pictures. So think of it as like you got this guy holding like this, and then this guy comes across it because of a hydrogen bond or an ionic bond. Or these guys are covalently bonded together by a disulfide bond. So now we're not talking about R groups that are right near each other. Now we're talking about R groups across the bigger polypeptide. Are we following? Okay. The quaternary structure was like this one down here. We have multiple polypeptides that got all folded up and nice, combined with other polypeptides that were folded up and nice, and together they work. Okay? So polypeptide is just a chain of amino acids. That chain, alpha helices or beta sheets from the immediate R group interactions, and then tertiary level with more hydrogen bonds, more ionic bonds, and disulfide bonds that are creating the bigger folding of that polypeptide. Multiple polypeptides come together for the quaternary structure. So I know we talked about it in chapter 2, but now we're understanding the bigger picture level of why that's important. Now, before we get into conjugated stuff, if I add heat, pH, which is a change in H pluses, so heat, we know we might see it as delta, right? pH, which is our hydrogen ion concentration, or other chemicals. Can you understand how that could affect our H bonds, our hydrogen bonds, our ionic bonds, or even our disulfide bonds if we have the right chemical there? We've already seen incineration, yeah? If you incinerate something, it turns it black to ash. We give off gases like carbon dioxide. That's because we're literally breaking down the covalent bonds in something, let alone those weak little H bond attractions or ionic attractions or disulfide bonds. We can break those with enough heat for sure. That's what incineration does. Or we can disrupt them with high or super high or super low pH, affecting how much H's are in there so that these hydrogen bonds here get disrupted. And so now instead of having alpha helices and beta pleated sheets and having our nice folded tertiary structure, we break down that structure and we revert back to just our amino acid sequence. Well, if our protein is normally shaped like this, like Pac-Man, and the thing that it binds on is shaped like a pizza slice. And now I have used heat or pH. Are we still shaped like that? No, because it was shaped like that because of our hydrogen bonds and our ionic bonds and whatever. So if we add heat, pH, or certain chemicals, that leads to denaturation. So are we tracking denaturation? That's what it is. We know we can denature DNA. If heat can disrupt the hydrogen bonds that hold DNA together, of course it can disrupt the hydrogen bonds that hold our protein together. So that's how denaturation can affect enzymes. So we're learning about that concept briefly in chapter 10. We will come back to it in unit 3 with chapter 11, because we're going to talk about microbial control mechanisms in unit 3. So understanding this basic here, unit 3, we're going to talk about chemical and physical methods of microbial control. And most of those are going to be denaturing protein or DNA. Because you denature protein, you're going to denature DNA too, right? Hydrogen bonds. It's just hydrogen bonds. Whether it's in DNA or protein doesn't matter. When you cook an egg, can you revert it back to the original raw egg? No, that's how serious denaturation of a protein is and how much it can affect. Yeah? Somebody's at your house, Barry. I'm assuming that's your ring alarm.
UPS.
Is it UPS? That's what my ring alarm sounds like. It depends. It depends on the level of denaturization. So we can reverse denaturation of DNA. That's how PCR works. But with protein stuff, it's a little harder. If it depends on how much you've denatured it. This is why having a fever at higher than 105 degrees is so serious, because once you get above 105 degrees Fahrenheit, now you're denaturing the proteins in your brain, and that can be irreversible. It can be. I'm not saying it always will be, but it can be.
And that's why you take Tylenol, like.
That's why you take Tylenol when you're pregnant. Exactly. Exactly, because you don't want that to happen. Anyways,
Just don't bring home one.
yes. Maybe following directions. Follow your doctor, not a president who's never had any medical training. Okay, so anyways, conjugated enzymes. Your conjugated enzyme is protein plus something that is non-protein. Protein plus non-protein. It really is that simple, okay? The protein portion is called the apoenzyme. The non-protein portion is called a cofactor. If that cofactor, so your cofactor is either inorganic or it is organic. If it is inorganic, we still call it a cofactor. This is like metal ions, like magnesium, which is necessary for DNA polymerase. DNA polymerase cannot function without magnesium. DNA polymerase is a conjugated enzyme. The protein portion of DNA polymerase is called the apoenzyme. The magnesium is the cofactor. Organic cofactors are called coenzymes. An example of this might be NAD or FAD. Or vitamins, vitamin C, vitamin B12, whatever. They usually work with proteins. If they're organic, I mean quite literally like organic molecules, carbons with hydrogens bound to them. Okay.
So.
What's the question?
One.
No, you're fine.
So are they in the cramp site on those, are they functioning as coenzymes? Because I didn't realize that.
They are.
They are.
They are.
Okay.
So we don't talk about that because it's too much, I feel like. I just feel like it's too much. I'd rather you guys understand the concepts of what is going on. So they are electron carriers. They are coenzymes, and they're working with enzymes throughout the whole process. So whenever we're converting glucose down to pyruvate, there's like a million steps in there, right? We're going from glucose 6-phosphate to like glutaraldehyde and like all of this stuff in there. And all of those intermediate steps that are in there, enzymes are guiding all of that. And so they are working with NAD and whatever, and then the NAD steals an electron off of something as a hydrogen carrying an electron with it, right? So that's what's really happening. But that's a lot. Okay, so all right, so this is the enzyme idea. This whole protein with its non-protein, once you are apoenzyme plus your cofactor and you're working in functional, we call that the holoenzyme. Holoenzyme. So DNA polymerase with its magnesium, holoenzyme. Okay, are we tracking that stuff? Conjugated enzymes. All right.
You could have a cofactor or a coenzyme, it doesn't matter.
Coenzyme is a cofactor. It's just an organic cofactor. So here's cofactors, right? Yep. If you are inorganic, you're still just a cofactor. If you're an organic molecule, then you get a special name, coenzyme. They're all cofactors. Yes. Yep, just rejoin it if you can. You'll have to use a different name if you do that. If you can, like I said. It's not that important as long as I got you guys participating in one question, but that's why I don't have the Kahoots as an actual grade, because it kicks people out a lot. Also, I'll remind you guys, if you guys are using OSU OKC as your Wi-Fi, do not. It is an old one. We don't support it anymore. Our IT program doesn't even support OSU OKC anymore. EDU Roam is the one they support. EDU Roam. You have to sign in with your email and your like login password for OSU OKC. Email is your username and then your usual like login password. EDU Roam. This one. Oh. EDU Roam. It's all one word, I think.
versus. Yeah, how long has that been going on?
Yeah, it's been for over a year now. That's been the case, yes.
I've been on Austin, OKC.
I know, everybody has, but like, that's part of the reason why you'll get kicked out. EDU Roam is a lot more reliable, but you have to log into it. So it's a pain, but
You know that you got
So
your
will it
password?
find it on the network?
it should. Especially like, once you log in once, it should repeatedly automatically
You know
join.
the username password for it?
It's your email and your login
Okay.
for OSU, like Canvas and
I
stuff,
can't tell you how many times I've gone to the library to like grind it out.
and you get
I-
kicked
I
out.
would get frustrated and just leave because there's no plan.
Try EDU Roam. All right, substrate of an enzyme. That's what I was gonna get into next, right? So like I said, we're gonna carry over a little bit into lab. We will survive it. Lab will be relatively, I say short, but we will not go over time, even with all of this, okay? I promise. All right, so if your enzyme looks like Pac-Man, and this is your normal enzyme, this pizza slice is the substrate. This is your enzyme. The site where it binds is the active site. All right. Way cool. All right, I know I haven't called on everybody, so I want to try to get everybody taken care of. All right, let's see. We have Victoria, Emma, Kristen. I feel like you already talked about stuff, didn't you, Kristen? I feel like you did. I didn't call on you, but I don't. Morgan, Shelby, Lily, and Cora. Any of those people, if you're online or in person. What is the allosteric site? Anybody know of those? Allosteric. It is a thing. It is this. It does. It does. So we'll start there. So any of those other people that I didn't call on, more specific than that, what does the allosteric site do? It does alter the enzyme shape, but in what context? I'll help you a little bit. Yes, exactly. It's the regulatory site. So Cora got it. It blocks the substrate by changing the shape of the enzyme. So basically, I'll show you what it looks like. We have two types of inhibition. Competitive and non-competitive. So I'm going to make this small. So Cora, you're good. This is our substrate. Sorry, that's our enzymes, excuse me. This is our enzyme. Normal. And then in our non-competitive, we will have an inhibitor that binds at the regulatory, also known as allosteric. Then when that happens, that causes our enzyme to change shape to like this. And again, our inhibitor didn't bind at the active site, did it? It bound at a different spot. And now we can't bind our substrate anymore because we changed shape. That is non-competitive. Why is it called non-competitive? Well, we have to understand competitive inhibition next. We didn't, we lost. All right, Emma, competitive inhibition. Where does the inhibitor bind? Competitive.
I repeat for nine. Where did it?
Yes, if we're an inhibitor. So this is two different types of inhibition. In non-competitive inhibition, we bound at the regulatory or allosteric site. In competitive inhibition, the inhibitor binds somewhere else. And where does it bind? So yes, that's good. So that'll count that because that does apply to what we're talking about here, okay? It binds at the active site. So if this is the inhibitor here, it mimics the substrate, so it's a different thing. So we call it competitive inhibition because it competes with the substrate for the active site. Make sense? Okay, so that's where I feel like that's the easy baseline stuff. It makes sense when an inhibitor binds at the place where the substrate binds and makes it so the substrate can't bind it. The more brain power one to me is the non-competitive, where you have the regulatory. So it's like if I normally carry my cup in my hands like this everywhere I go in order to do my work, well, if you come in and you put, I don't know, hand sanitizer in my hand instead, how am I going to carry my cup? That's competitive. We're competing for the same spot. But now you say, okay, you want to carry stuff with your hands. Now I'm not going to adjust your hands. I'm going to tie your elbows behind your back. How are you going to carry your cup now? That's non-competitive. So we're not affecting the hands. We're not blocking the hands. We're changing the shape of the protein so that the hands don't work anymore. So that's the idea of competitive versus non-competitive. All right. Okay, cool. Okay. All right, so I'm going to clear the screen. Do we have our notes down? All right, because you know what's probably coming next. Girl drawings. I want to get everything clear before we get into the next stuff. We're almost there. Okay, what causes a denatured protein? We talked about this a second ago. We still got Morgan, Shelby, and Lily. I feel like Kristen participated with discussion, so I'm good with it. Okay. pH, heat, and chemicals, which we talked about previously. We're good. It changes those secondary and tertiary structures. What best describes non-competitive inhibition? We just talked about it. The inhibitor, what's it doing? In non-competitive. Go away. So it attaches to the allosteric, also known as the regulatory site. Morgan, you hear Morgan. This one attaches to the active site. What type of inhibition was that? Competitive. Okay, just a review, just to say it and make sure we are tracking with that. Okay, excellent. I know what time it is. Okay, because we still got to do glycolysis and all that. And I know you're like, oh my gosh, it's so much. It's less than you think. Okay, I promise. The 5-carbon sugar found in ATP. So a little bit with this. This is a picture of mitochondria, by the way. We have a 5-carbon sugar. We have adenine as our base. And we have phosphates, PO4, PO4, and PO4. All right, so this sugar, 5-carbon sugar, is ribose. So remember that ATP, you guys remember what a nucleotide is, right? It's a sugar, a phosphate, and a base. So is ATP, but instead of 1 phosphate, it's got 3. So that's the only difference. It is a nucleotide. It's a ribonucleotide, like we have in RNA, but with 3 phosphates. Okay, so how is energy stored in ATP at the chemical level? What is why does this have energy? How is this energy? Does anybody know? Not, that's how we'll make the ATP, but ATP now is our energy molecule that we get from that. Tawny. So each of these phosphates, y'all, we say PO4, but what is a phosphate really? POOOO, negative, negative, negative. Now on a phosphate here, that negative is attached to something, but at least one of those oxygens chilling out. This one will be attached to something, this one will be attached to something. But this oxygen on all of our phosphates, PO4, is negatively charged, negatively charged, negatively charged, and this guy has 2 negative charges on him. Now, you guys ever played with magnets? If you put 2 south poles together, what do? Push apart. It's the same thing here. Opposites attract. Negative likes to be by positive. So we have covalently bonded, covalent. Oh, here's a C for you. 3 negatively charged things in those 2 covalent bonds. Holding a south pole covalently to a south pole, covalently to a south pole. They are pushing each other apart and they hate being held together. So the way that we get our energy from them, we cleave off one of those phosphates. And when we do that, that helps us power things in the cell. The way that we actually chemically do that, that phosphate will either be removed entirely and it will cause something else to happen, or we'll move that phosphate onto a protein. Now, we talked about how a lot of our proteins being folded the way that they are, they're folded that way because of ionic bonds, hydrogen bonds, charges, interaction, and attraction forces. So if I put a negatively charged phosphate onto a protein, it's more than likely going to cause that protein to change shape, right? And that changing shape can drive chemical reactions. That is how the cell gets energy from ATP, usually by using those phosphates that are attached to it and putting them on other things. You following? Okay, other chemicals or proteins. So that's ATP and how it works. So now we remember why ATP is so important. Now we can talk about glycolysis and all of that. Okay, because the whole point of glycolysis was to make ATP. Not glycolysis, but all of aerobic respiration. Okay, so there are 3 types of respiration. There is aerobic, anaerobic, and fermentation. Aerobic respiration consists of glycolysis. We'll talk about what they are in a minute. I promise it's going to be shorter than you think. Glycolysis, Krebs, and the electron transport chain. In aerobic respiration, the final electron acceptor is oxygen. In anaerobic, we have glycolysis, Krebs, and the electron transport chain. It's the exact same, except the final electron acceptor. In anaerobic, it is anything but oxygen, okay? It could be sulfate, nitrate, nitrite, all sorts of stuff. Not oxygen though, okay? Period. In fermentation, it is just glycolysis. So let's talk about these things. Glycolysis. In glycolysis, we are taking a 6-carbon glucose, which remember is C6H12O6, and turning it into 2. Let me give it a little more space. Hold on. Turning it into 2 3-carbon pyruvate. Notice how we went from 6-carbon to 6-carbon. All of our carbons are intact. Do we make carbon dioxide in glycolysis? No. We know that because all of our carbons are still there. We literally just took a 6-carbon thing and split it into 2 3-carbon things. We didn't lose any carbons. carbons don't spontaneously generate. They had to come from carbons. Okay, so in glycolysis, we make products. We do. We make NADH and a little bit of ATP, 2 net. But what is the point of glycolysis? What is the total goal of glycolysis? To make pyruvate. Do we get some ATP? Yeah, but it's very little, only 2. So we got a little 2 ATP. We got some NADH. Now, why is NADH significant? Why am I even listing it and who cares? We said it's a coenzyme because enzymes are working with NAD and we're adding on an electron with it. So I want to be very clear. This is NADH, that hydrogen, just like when you have H2O. When we are covalently linked to oxygen, we are sharing electrons between the hydrogen and the oxygen, right? That hydrogen has an electron that came with it. So does NADH. So this NADH actually represents an electron. Okay? We tracking? NAD, no electron. NADH, electron now. So it's an electron carrier. So it really represents an electron. So we got ATP, cool. We also got we got an electron in NADH. It's an electron. If we are going through fermentation, in fermentation, we stay here. If we are fermenting, we stay here. We can't keep that electron. Can't just have electron collecting and not getting used and just hanging out. So what we do in fermentation, again, this will be in the red. This only applies to fermentation. We will feed that electron back into pyruvate and then turn pyruvate using that electron into lactic acid or ethanol and or carbon dioxide if we feed that electron back in, only with fermentation. So this allows, if you're going through fermentation, you can still get some ATP. It's going to be very little, only 2. But it allows you to get some ATP in the absence of oxygen. But we still have to deal with the electron. We just feed it back into pyruvate. And as we do that, it changes pyruvate into lactic acid and or ethanol and or carbon dioxide. There are other acids that can be made too, but these are the gist of it. We're going to be seeing this a ton in the second 8 weeks of our lab. I mean, almost every lab will be that. It's fermentation. Okay. So this is, again, another bread and butter that doesn't feel like it's going to be a bread and butter, but it is going to be. Okay. Okay. We're tracking what's going on with fermentation before we move on. That's just fermentation in the red. In our regular cells, the goal is to make, in aerobic respiration, the goal is to make pyruvate. Okay. The goal is pyruvate. That's it. You're welcome that I'm not making you know glucose 6-phosphate and BBBS after that. Because this is hard enough, right? The next step is that intermediate step. The goal of the intermediate step is to make acetyl-CoA. Acetyl coenzyme. Coenzyme. You guys know what a coenzyme is. It's an organic molecule that binds with enzymes to help the enzymes work. Acetyl coenzyme A. So this is taking 3-carbon pyruvate and turning it into 2-carbon acetyl CoA. Each pyruvate will do this. Well, we lost a carbon in there. So what's it going to come out as? Carbon dioxide. We'll make a little CO2. We'll also make some NADH. So we also make NADH, which is an electron, and a little bit of CO2, which is going to come out as waste that you exhale. Where does your carbon dioxide that you exhale come from? The food that you ate. Yeah, from glucose, whatever. Pyruvate, but it used to be glucose. Okay, so we finally gotten there. All right, now we're in the Krebs. Yes, did you have a question?
2 carbons pyruvate
Mhm.
and then 2 carbons the carbon dioxide and the NADH, are those all 3 of those things the products of 3 carbon?
So we would say pyruvate, and the products are carbon dioxide, NADH, and acetyl-CoA. Mhm. But the main thing that we care about is acetyl-CoA. The whole goal. We'll keep those guys in orange, right? I don't know if it looks orange to you, it looks more yellow, I don't know. Anyways, so we're in the Krebs cycle now. We will take our 2 carbon acetyl-CoA and we will add it to a 4 carbon molecule. It doesn't matter what it is. It happens to be oxaloacetate or oxaloacetic acid, interchangeable. If you're like, you're saying pyruvate, it's pyruvic acid, go check yourself before you come have this discussion with me, because in the body, those are interchangeable. Sometimes it acts as an acid, sometimes it acts as ate. It just depends if it has its hydrogen bound to it or not and how it's interacting with molecules. They're the same. Ate and acids are the same in the body, okay? So pyruvate or pyruvic acid, oxaloacetate or oxaloacetic acid, they're the same, okay? 4 carbon thing, it's gonna make a 6 carbon thing. That happens to be citric acid or citrate, however you wanna pronounce it. Happens to be, which is why we also call the Krebs cycle citric acid cycle. But the important thing is that we're taking our 2 carbon thing, making it with a 4 carbon thing to make a 6 carbon thing, putting it through some steps that eventually regenerate the 4 carbon thing, which will then link with a new and keep the cycle going. So, and we're kicking off in the middle. If we're going from 2 plus 4 to 6 and then turning it back into 4, what are we kicking off in the middle? Carbons in the form of carbon dioxide. So we'll kick off a couple carbon dioxides, a CO2 here and a CO2 here. So we know we're kicking 2 of those off because we got to regenerate our 4 carbon thing. Okay. While we are kicking those off, what are we making? We might make 1 ATP. Who cares? We might make an ATP. We're going to make some NADH. We're going to make some NADH. It doesn't matter where or how. I don't care where in the cycle that happens. Big picture it for me, okay? We're going to make some NADHs and we're going to make some FADH too. Okay, what is the point, the goal of the Krebs cycle? To make NADH and FADH2. Why? Because these carry electrons. The point of the Krebs cycle is to collect electrons. Do we also make a little ATP? Yes. Do we also make carbon dioxide? Yes. But the point of it is to make electrons. So glycolysis, we are taking glucose and turning it into pyruvate. That was the point of glycolysis, so that we could turn pyruvate into acetyl-CoA and turn that into electrons. That's it. Do we understand why the electrons are going to be important? I hope that we do, because that's the electron transport chain, right? Okay, so we saw our picture here with respiration, our different levels of respiration here. Now I'm going to erase that so we have room for our electron transport chain. This is our last part. And we'll briefly touch on photosynthesis. Any questions so far? I honestly, when you guys were learning about this in chemistry, don't you wish that somebody told you the big picture of it first before they asked you to memorize oxaloacetate and glucose 6-phosphate and blah, 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 which I feel like you get lost in all of those little steps. You don't see why we're even doing this or what the big picture is. The whole point of breaking down glucose is to get pyruvate, so you can't feed it into Krebs and get electrons. That's it. That is it. That's the whole point of those guys. All we need is the electrons from the Krebs cycle. Now we can go into the electron transport chain. And I know the book calls it the electron transport system. I don't care which one you call it, but on the test, I'll almost exclusively call it the electron transport chain. They are the same thing, okay? So the electron transport chain looks like this. You have a membrane, and then we have some proteins next to each other. We'll put 3 of them, who cares? And then we will have a bigger protein that looks like a channel. And this is ATP synthase. All right. We know that the membrane, it goes on, and then these are just repeated over and over again. Now, this membrane setup in bacteria, this membrane setup is on the inner membrane or cytoplasmic membrane, whatever you want to call it. Of the cell. If you're a gram negative, we're talking about the inner membrane. If you're a gram positive, it's the only membrane. Okay? So it's along that. It's like on here, and then ATP synthase, all along that inside membrane, okay? In eukaryotes, you have the whole big old cell, and inside of that cell, yes, you have a nucleus, but you also have these structures that we call, it's not this big, but I'm drawing it for just drawing purposes, mitochondria. And inside of that mitochondria, they have an inner membrane. Let me draw it better. Let's draw it in a different color. Pen. Let's draw it in, I don't know, blue. Their inner membrane of the mitochondria is called the cristae. So all of this is set up on whatever. That's our stuff on the inner membrane. You see how it's the same thing though? Because we agreed mitochondria basically used to be bacteria. They have their own genome, stuff like that. So are we seeing how this is set up differently? Okay, so now we can talk about our electrons and everything like that. Like I said, we're almost done, I promise. Okay, NADH and FADH2 carry with them H's with electrons. So they're going to give off an electron or 2. And they're going to give off also the H's that were attached to the electron as H pluses. Right? Because the H's are together an electron and an H plus. So now when the electrons literally separate from that, the electrons move through these complex of proteins. These proteins, each time that we move through there, again, negative charge moving through proteins that are held together by attractions causes the proteins to change shape. When they do that, it pumps hydrogens out. So these negatively charged electrons are going to move through these proteins. These are called cytochromes. When the negatively charged electrons move through there, it causes the H pluses to be pumped out. So we'll collect a whole bunch of H pluses. Right? We'll have a whole bunch of them. This whole thing with all these H pluses is called the proton motive force. Now, if we are building up a whole bunch of bouncy balls that are held back by the membrane and physics says they want to equal out on both sides, some of those H pluses want to come back in, right? We got a whole bunch of H pluses on the outside, we're forcing them out. And now they want to come back in. The only way they can come back in is through ATP synthase. So we're going to get some H pluses come back in. We're going to have these electrons moving down here, but they can't stay at the end of the cytochromes. And they don't just disappear into the ether. They have to go somewhere too. So as the electrons come through, we're going to take an electron with each of these H pluses. Whoops. Wish there's an undo button. Electron with the H pluses. And we're going to take half of a water molecule. What's half of O2? Just an O, right? Now imagine we're taking this and this and this. What does that look like? H, H, and O, right? So to get rid of all the H pluses so that we don't want them building back up in the membrane. Otherwise, you're going to equal out on both sides of the membrane and you can't keep that gradient going. We want the H pluses to keep coming in, but they're only going to keep coming in if we get rid of the ones that came in. So that is going to be what we breathe in oxygen for. This is the literal only reason in your entire existence that you breathe in oxygen. This is the only reason you breathe in oxygen. You have to breathe in right now and you exhale carbon dioxide. You breathe in the oxygen here to keep that proton motive force built up. You exhale carbon dioxide because you made it as waste in your intermediate and your Krebs step. Those oxygens and carbon dioxide never touched or interacted with the oxygen that you exhaled other than they mixed as gases in your lungs. They're not chemically interlinked. Tell me it's not the coolest thing ever to see it and know why you even inhale or exhale. And that if I suffocate you right now, the reason you die is because you stop making ATP there. That's it. The only reason you inhale oxygen is to keep this going. That's how much your body relies on the ATP made by aerobic respiration. So good. Okay, so obviously this, when we bring our H's through, we're going to make, and I'm leaving it out like it's some little thing. We're going to turn ADP plus some inorganic phosphate and turn it into ATP. This is obviously the goal of the electron transport chain, but I can't stress enough the importance of keeping that oxygen inflow in so that we can turn it into water. But we're just getting rid of the electrons and the protons so we can keep the whole thing going. Any questions? So yes.
So after it comes, the nitrogen comes through the ATP synthase,
Mhm.
does the negative electron just kind of stick with it? Or what is that electron?
So that last cytochrome, that very last cytochrome is called cytochrome C oxidase. It's an enzyme. And so that enzyme works with that last electron and works with the oxygen coming in. It's a little more complicated than this, right? But using ATP synthase and that cytochrome C oxidase, they work together with the oxygen to use the protons and that last electron together to turn it into water. But cytochrome C oxidase is the one that's going to be doing that. Okay, so it's the last cytochrome. I'm not going to test you on that in here in the lecture. That's a little more above our pay grade for the lecture. But when we get to our very last lab in all of the labs, it is our respiration lab. And I do expect you to know that it's called cytochrome C oxidase because we'll be testing for literally cytochrome C oxidase in that lab. Okay, so it will tie together in our last lab, like quite literally our very last lab. Okay, any other questions about that? Okay, so that is what's really going on. Glycolysis is turning glucose to pyruvate. Intermediate step turns pyruvate into acetyl-CoA. Krebs turns acetyl-CoA into electrons. Electron transport chain turns those electrons and the protons into a gradient that allows us to make ATP. Oxygen we breathe in to keep that gradient going. That's it. Okay, we're good. That's the big picture of it. All right, so remember, plants do this too. in photosynthesis, okay? I know it. We're almost done. In photosynthesis, we will take sunlight on a membrane where we have other pigment molecules called chlorophyll. And then they'll have another, looks like ATP synthase because it is, H pluses. Chlorophyll excites this stuff, excuse me, sunlight excites the chlorophyll. Excited, excited, excited. This causes H pluses to be pumped out. We get a ton of pluses out here. Then the pluses come back in, exactly like up there. But instead of driven by electrons, it's driven by sunlight exciting everything. We'll make some ATP in the light reactions. Then we will take that in the dark reactions, also known as the Calvin cycle. Then in that part, we will take that little bit of ATP plus carbon dioxide. And again, I don't need you to know all the steps, but we're going to turn that ATP plus carbon dioxide into C6H12O6 glucose. This is what makes them autotrophs. They can take carbon dioxide inorganic and turn it into glucose. And then what do they do with that glucose? They can use it to build molecules of the cell, but they can also feed it back into glycolysis, Krebs, and the electron transport chain to make more ATP. Now we only made 2 ATP in glycolysis. We'll make 2 more in Krebs, 1 for each acetyl-CoA. Okay, so you get 2 ATP from a glucose in glycolysis. You get 2 ATP from a glucose in Krebs, because we go through it twice. And then how many ATP do you get from the electron transport chain? You get like 32, something like that. I don't even know the exact number. You can sit me here, be standing here being like 2, 2, 4, or 34. This is 38 overall for bacteria and 36 overall for us. Why would it be less for us than for bacteria? Because we have glycolysis and Krebs in the cytoplasm, and then we have to get stuff into the mitochondria for the electron transport chain, and that costs us energy. So we lose 2 ATP because we're moving into the mitochondria. So 34 ATP made in the electron transport chain, that is the goal of the electron transport chain. ATP. Okay, questions about this? We're good? I know it's a lot. I know I did not call on everybody, but that's okay. We had a good discussion today. So if you were here today, I'm giving you credit. Okay. If we didn't get to you, because I know what we're going over time. And so if there's no more questions on that. We're going to have a 10-minute break. I say at 11:30, 6-minute break. 11:30, meet me up in the lab. Okay.